Well, good morning, everybody. Today I'd like to welcome you to the Michael Lecture. Dr. Michael was an important pediatrician in our area, and for his 90th birthday, his daughter, Robin, who's here, and his son-in-law, Scott, who is here, and other members of the family created the Michael Professorship to honor him. And for a number of years, he actually was with us to be part of Grand Rounds. Unfortunately, he's no longer with us. But I would like to invite Dr. Koenig, Dr. Michael's son-in-law, to come and talk to us a little bit about Dr. Michael and what the goals were of this Grand Round. And Dr. Koenig is our past chair of the CRI board and is the CEO of Macrogenics, which is a major biotechnology company focusing on immunotherapy of cancer. And he was just a wonderful chair for us and has continued to work with us in developing commercialization. Dr. Koenig.
Well, thank you very much, Mark. It's a real pleasure to be here this morning with Robin. First of all, to be able to hear Mark as the lecturer this year for the Michael Lecture. Mark, my incentive to actually be part of the children's family was really a lot of what I saw in Mark and his leadership from both academic as well as research initiatives, and it's been really motivating me and the family over the years of continuing to sustain this lectureship. Just a little bit about my father-in-law, Morris Michael, who was known as Mike, would have actually, on June 8th next month, would have been 99 years old. He passed away 6 years ago. He was born in Bessemer, Alabama, moved to Kingston, New York, and grew up there, was essentially a scholar of the family, went and got his education at Cornell on a full scholarship, and then moved to the Washington area as a medical student at GW. He graduated second in his class, and then did his internship at the district General Hospital before he was called for service into the Army. After the Army, he was trained in the treatment of children at Johns Hopkins before there was really a formal pediatric residency program. He was part of the infectious diseases department. He established an office over on Cumberland Street off of Connecticut Avenue in his house, and maintained his practice for over 40 years, and was an adjunct professor over at Georgetown. He has had historical affiliations with children, and actually was honored at Georgetown as a pediatrician of the year one year. What was very inspiring about my father-in-law is his love for learning. Even after his retirement, he actually attended Grand Rounds, as Mark was pointing out, even coming to these rounds on a weekly basis. In fact, in his enfeebled latter years, my wife would take him to the Grand Rounds on a weekly basis so he could enjoy that till his death. We established this 10 years in his name and also in my mother-in-law's name, who helped to support him over the years, Mildred Michael. The idea here was to perpetuate the knowledge, particularly in the area of biotechnology and cutting-edge science. So it's really an honor to hear today from Mark on the rebirth of gene therapy, an area that I also have interest in, and so I look forward to hear. And a little birdie told me, somebody by the name of Naomi, who had previewed your lecture, that it's really good. So I look forward to it today.
Thank you. Well, I'm just back from the pediatric academic society meetings where children's was extremely well represented. We gave over 150 talks, posters, workshops, special interest groups, probably the most of any other institution. We compared ourselves to Boston Children's in terms of the number of presentations just by our residents, and they're a much larger resident program than we were, and we matched them in terms of the number of presentations. And also, you know, there are 4 societies that that run the Pediatric Academic Society meeting. There's the American Pediatric Society, the Society for Pediatric Research, the American Academy of Pediatrics, and the Academic Pediatric Association. Well, it turns out that we have, this year, we were leaders of 3 of those 4 societies. So Mario Ottolini was president, or is president, of the APA, and Anna Penn, who is one of our neonatologists, is the secretary for the Society of Pediatric Research, and I was the president of the American Pediatric Society. So it was really a great year for Children's National there. What I'd like to do this morning is, in keeping with the Michael lectureship, is to talk about transformative research and how we can really change children's lives through transformational change. But at the same time, this is a cautionary tale. It's a personal tale. It's about my own research career and how it was impacted by the death of a patient in an early gene therapy trial, and what I learned from that experience. And it's especially meant for those of you who are beginning your career or early in your career, because it shows the importance, especially for those of us who are clinical trialists, to practice clinical trials in the utmost careful manner. But it also shows that even when you make a major error, as I did during this trial, that there is a possibility to recover from it, and the resiliency that we all must have during our career in practicing medicine in an institutional setting. And I chose the title of this, The Phoenix Rebirth of Gene Therapy, just to remind you that mythologically, the phoenix was a long-lived bird, and it lived so long because every time it died, it arose from the ashes to be reborn. And that really is what happened to the field of gene therapy. I'd like to acknowledge at the beginning those who have been very involved with me in producing the science that I'm going to talk about later in this discussion. I've worked for almost 30 years now with Jim Wilson at Penn, who is one of the pioneers of gene therapy, a virologist and a physician. And Lily Wang is his second in charge and is a wonderful, wonderful scientist. And Peter Bell, a pathologist there. And here at Children's National, my laboratory partner has been Hiroki Morozano, and Mendel has been very helpful as we've designed the clinical trials for the disorder. And Bob McCarter has been not only our biostatistician, but has been extraordinarily helpful in actually figuring out with us how to design the study and how to interpret the results. We've been funded both by the NIH, by program project grants, and also by family, particularly the Kettering family who has a child who has a urea cycle disorder, which will be the focus of this talk. And I have no conflicts of interest to report. So I am going to talk about the urea cycle. When in the early and mid-1970s, I was a trainee at Johns Hopkins, and this was only about a decade after these rare inborn errors in metabolism were first described. For those of you who may not be familiar with urea cycle disorder, you probably remember from medical school the urea cycle, and that this is necessary for the breakdown of protein and to make sure that waste nitrogen that comes down from the breakdown of amino acids that are not used for enzyme hormone production are further broken down, and the ammonia that's released is converted to urea through this multi-step pathway that it occurs in the liver. However, there are children who have been identified with blocks in any one of these urea cycle enzymes, and together they represent about 1 in 40,000 children. So it's a rare disease, one of the rare diseases that Marshall Summer is involved with in the Rare Disease Institute. They're all inherited as autosomal recessive disorders, except for ornithine transcarbamylase, or OTC, which is the most common and is inherited as a sex-linked disorder, so that the males tend to be more severely affected and present in the newborn period, and the females are heterozygotes, carriers, and about 15% of them will present as a late onset disease. And so I identified a 13-year-old girl who had never had the diagnosis as having had urea cycle disorder, and working with my colleague Saul Buslow at Johns Hopkins, we developed a series of novel treatments that have been called alternate pathways for waste nitrogen excretion, basically creating detours around the enzymatic block. And this really converted these group of disorders from fatal disorders to chronic diseases that could be treated. And this was an article in the New England Journal of Medicine from the early 1980s where we reported this and the outcomes of that. Well, shortly after that, I was recruited to CHOP, and I went there, and that's where I met Jim Wilson, who had just come from the University of Michigan. And I went up to him and I said that I was very interested in working with him on developing gene therapy for urea cycle disorders. And we chose ornithine transcarbamylase deficiency to be the disorder that we wanted to focus on for a number of reasons. First of all, although it's a rare disease, it was common enough that there were a large number of children out there, about 2,000 in the United States, who could benefit from this. It was the enzyme, as I mentioned earlier, is located in the liver, and the advantage of that was that the primary mode for doing gene therapy at that time was using an adenovirus vector. So you basically are taking a virus, and a virus is just DNA with a protein coat around it, the DNA viruses, and basically cutting out a piece of that DNA and inserting the DNA for the missing or defective gene, and then reinfusing that into the patient or a mouse model to begin with. So the adenovirus had an affinity for the liver, so it looked like it would be a good thing to use for OTC deficiency. The clinical presentation was very severe, hyperammonemic coma, and so that if we could use a fast-acting adenovirus, we could both prevent brain injury and prevent death from occurring. And although we had developed the alternate pathway therapy I described, it was incompletely effective. took a while to last. It was stoichiometric, which means that it could only remove a certain amount of ammonia, and if these children had ammonias of 1,000 micromoles per liter, where normally it's less than 30, it was not effective until you got it down to maybe 300. So you had to use hemodialysis for that purpose. The gene therapy would have replaced it, and if it worked fast enough, could have gotten around this problem. And the outcome was still not optimal. Most of the children who survived had intellectual disability. And finally, there was a representative model called the sparse fur mouse, or SPF-ASH mouse, which had a partial deficiency of OTC with about 10% activity, so we had a good model that we could work with. So that's what we decided we were going to do, and we started doing this in 1989, and it took us about 10 years to go through what's in this slide, which is first of all to develop an adenovirus that really could be effective, then to try it in the sparse fur mouse, and then to take primates and do safety studies on it to make sure it was safe in primates, and then finally to try using it in humans. And so in 1998, two things happened. First of all, we opened the study at the University of Pennsylvania, and I moved here in my current role. But I continued to be one of the 3 investigators for this study. And the study was basically set up as a phase 1 safety trial. And with that, what you basically are doing is taking about 3 patients in each group and do progressively higher doses of the gene therapy. It would be an injection directly into the liver. And the highest dose, so we did 6 doses, and the highest dose was the dose that would be predicted to be effective based on our preclinical studies in the mice. And things went well until the second patient in the 6th cohort, the highest cohort. And this was a patient named Jesse Gelsinger, who was an 18-year-old young man from Arizona who had partial OTC deficiency and who had had recurrent episodes of hyperammonemic coma. And in fact, a year before this happened, he had been in coma, and the alternate pathway therapy we developed had saved his life. And he decided, he was a very altruistic young man, that he wanted to do something in return, so he participated in the study. And within 4 days, he was dead. He developed what's called systemic inflammatory response syndrome, which is illustrated here, where he developed fever, coagulopathy, went into coma, developed adult respiratory distress syndrome, and SIRS has a very high mortality rate. Subsequent studies that we did indicated that he developed the systemic inflammatory response syndrome as a result of an immune response to the capsid, to the protein coat of the adenovirus. And this led to not using adenovirus anymore for this type of therapy. Well, you know, that in itself was devastating to the family, to us. But in addition to that, the FDA came in, and in looking at the way we had performed the trial, found a number of errors that we made, which made it even worse. And it turned out that this was then devastating for the entire field of gene therapy. And over the next decade, there was very few trials performed in gene therapy. There was the money dried up for the use. So this single event, you know, had this effect, and it was an event that was really throughout the media. This is an article that one of the main articles in the New York Times magazine a couple of months after Jesse died, and many of you may remember all of this going through. And it was a a very difficult time. And when the FDA came in, they identified 5 major mistakes that we have made. And I want to go over them with you so that those of us who continue to do clinical trials can be careful about each one of these things and can learn from them. The first was the area of conflict of interest. One of the 3 principal investigators had a significant financial interest in the outcome of the study. He had equity interest in a drug company that, if this had been successful, would have been helpful. We recognized this, and University of Pennsylvania had developed a management system for this potential COI so that this investigator had nothing to do with the performance of the clinical study. But that barrier didn't hold up once a patient died, and in fact has led to changes in both institutional regulations here and regulations federally, where if you have a significant financial interest in the outcome of a study, you can have nothing to do with that study anymore. So that was the first learning. The second is the issue of informed consent and the risk of influencing patients. And here we were criticized because this was a rare disease. The issue was because I was the metabolic physician for a number of the patients, I didn't participate in the obtaining of consent because I was worried that I might influence the patient. You know, the patient inadvertently, just like we have unconscious bias, you could also have that in regard to the study. If you think the study is really important, you may present it in a way that is more positive and emphasize the benefits and perhaps not emphasize as much the risk. So I didn't participate in the study, but the concern was that even though I didn't participate in the consent procedure, that it was well known that I was enthusiastic about the trial, and therefore would patients have been influenced to participate because of that. That's a more difficult thing to get around, but it's one that we really need to be aware of. The third was amending the consent form. So in our consent form, we had said that there was a possibility that there would be liver dysfunction after gene therapy was performed. And in fact, after the first 2 or 3 patients, we observed that there was a transient elevation in liver function tests and then went down. And so what you have to do in that case is change the consent form from saying there's a possibility of it to saying we have observed, 2 to 3. And we waited for the annual report. Each year you have to update your consent form. And that was an error because whenever you find something new in terms of a change in adverse effects, you need to change that in a very brief period of time and not wait for the annual review. So that was another error of ours. Assure adherence to protocol. This was perhaps our largest error. We had indicated in our protocol that we would not treat a patient, they would not receive the vector if their ammonia level is above a certain level. And his was above, mildly above that level on the day of the trial. We gave him his regular medication as was on the trial, and at the time that we infused him, the ammonia level was normal, was back to where it ought to be. But because in our protocol we had said that on the day, if it was above a level, we shouldn't have done it. We shouldn't have enrolled, we shouldn't have done the study on that day, but we did. Even though the elevated ammonia level has nothing to do with development of SIRS, because we didn't follow protocol, that was the biggest error that we had. So it's really important that you follow protocol and that clinical judgment doesn't come into play when you're doing a clinical trial. And then lastly is the need for effective team organization. So we had 3 principal investigators in this study. One who developed the virus and was responsible for making the vector and making sure it was working well. The second, who was me, was the metabolic physician to make sure that all the metabolic parameters we were studying were done correctly. And then the third was a surgeon who actually ran the clinical trial and infused the vector. And the result of that was that we divided up responsibilities, including the responsibilities of reporting to the FDA and compliance and regulatory issues. And what we found in retrospect was that there were areas of overlap between what the 3 of us were doing, so that we were delayed in putting in certain required information. And it was missed because, one of us thought the other one was done. The lesson here is that although team science is absolutely essential, when you're running a clinical trial, there needs to be one person who has the ultimate responsibility, who's going to run that trial and make sure that all the regulations are taken care of. So for me, someone who had considered himself an experienced clinical trialist, this really involved a tremendous effect on my own self-image of what I was as a scientist, as an investigator. And I took additional training to remediate my knowledge in clinical trials, to learn about human subject protection in a way deeper than what I had before. And because this was a year after I had come here in my role as Chief Academic Officer, I instituted a number of things at Children's National to make sure that the mistakes I made were unlikely to occur at our institution through training. And we really have had an exemplary record due to all of your efforts and to the efforts of our IRB and other regulatory areas. Recently, I asked my wife, Karen, what it was like living through this period. This is a picture of my wife and our 3 children at the time. They were young adults in 1999. And she said that I had always shielded my family from what was happening bad at work so that they made it through it. But she did say that I seemed very stressed and anxious, but then she also said that's the way I usually am. So we lived through it as a family. I lived through it as an individual. I grew with it, but there hardly a day goes by that I don't think of Jesse Gelsinger and his family, and hope that the work that we continue to do, and which I'm going to describe next, will honor him by eventually finding a cure for urea cycle disorders. And what we have focused on has been adeno-associated virus gene therapy. Now, although it sounds like adenovirus, it's called adeno-associated because it was found to be that the AAV virus was found to be close to the adenovirus. The adenovirus is this big, the adeno-associated virus is very, very small. But this is a picture of it, what it looks like, and it has at least 3 real advantages. One is it's quite small, so it can get in to places, and it can go to many different organs depending on what the tropism is of the particular type of the virus, and you can make this virus in many different ways. The second is that unlike the adenovirus, which had this terrible immune response, the AAV doesn't appear to have that response. And as I mentioned, there are many serotypes, so that you can have different tissue tropisms. Well, up to this point, there have only been 2 gene therapies that have been approved for use. Both of these have been in Europe. And the first one is Glybera, or Uniquir, this was approved in 2012 and became a commercial product in 2015 in Europe. And Uniquir is a treatment for lipoprotein lipase deficiency, or LPL deficiency. This is a cause of severe pancreatitis. And this is an AAV1 vector that is infused into the muscle, because the AAV1 has an affinity for the muscle. And here you can see in an animal model of the LPL deficiency, this is blood or serum taken from an animal who has the disorder, either untreated or treated, and you can see the difference of how it's cleared by the gene therapy. The second compound that has been commercialized in Europe is a treatment for adenosine deaminase deficiency, a form of severe combined immunodeficiency. And this is individualized for each patient, where you basically take out hematopoietic cells, you isolate CD34 cells, you then take a retrovirus and put the retrovirus that contains the ADA gene into those cells, and this is all shown here, and then it is infused back into the patient. And both of these have been successfully used in Europe. In the United States, the gene therapy that is closest to coming to fruition is the use of AAV2, which is eye-directed for the treatment of Leber's congenital amaurosis, which is a form of retinal dystrophy in children. And this work has been led by Dr. Bennett, who I know well at the University of Pennsylvania. It just shows you the length of time it takes to develop this. It's been 20 years since initially it was shown in 1994 that you could take a viral gene and transfer it into the mouse eye, and then showing it in the monkey, and then in the canine model, and then it's been 10 years since the first safety study was done in humans, and it's likely that in the next year or two this will become commercially available. And I should mention that the cost of a single gene therapy injection will probably be about $1 million. So if we look at the number of gene therapies that are being done for all different disease groups, you can see that by far over 2/3 of the more than 1,000 trials that are currently involve either cancer or rare diseases. And if you look at the number of gene therapies in active development, you can see my point earlier that for a period of well over a decade, there was no growth in the development of gene therapy. It's really only been in the last 2 or 3 years that this has happened. And this very rapid growth has been due to a number of things. One, the development of the AAV vector. The second, the improvement in the economy, and therefore increased investment by foundations, by the NIH, and by industry. And for us, at least, we have had NIH funding for the last decade, actually now, to develop this using AAV gene therapy. And the reason we use the AAV, and this is just a cartoon of it, so here you have the DNA, you have the protein coat here. This is the DNA, and basically what you do is you put the OTC gene in here, and then you put a promoter, in our case it's called a TBG promoter, which basically is the GPS that gets it into the liver itself. And the goals are to develop a safe capsid, to use a single-stranded DNA instead of a double-stranded, because it then is transduced and can be expressed much more rapidly, which is what you would need in an acute disease like urea cycle disorder. And then to promote it, have a promoter so that it goes directly into the liver. And then it took us a lot of time to actually optimize the gene that we were putting in, the cDNA, for that. But we finally were able to get a version that we thought would work after many different trials. And this is number of weeks after treatment. And orotic acid, urinary orotic acid, is a biomarker for OTC deficiency. If you don't have any OTC, the orotic acid excretion in urine is very high. And this is the normal range in this shaded area here. And you can see the levels are very high to begin with. And these are the different doses of the gene therapy that we gave. And you can see that within a week or so, the levels came down to normal, and they lasted for close to a year. So we basically were able to cure, at least for a year, the mouse, the sparse-scratch mouse, which had partial OTC deficiency. On the bottom here is an immunostaining for the OTC enzyme activity itself. So you can see it's quite dark in the wild-type normal animal, and there's almost nothing there in the sparse fur animal. And at 23 and 30 weeks at the various doses that we gave, you can see that the levels are really quite close to the wild-type. So we really had shown that this could be effective, and this has actually led us to propose a clinical trial in patients with partial OTC deficiency, and this is an industry trial that Children's National will be part of, although I won't be a principal investigator with it. It will be directed by Nicholas Amu, who's one of our gifted geneticists and metabolic specialists, and will be starting later this year. But we also wanted to see what we could do for the neonatal onset disease. This is still the most lethal form of the disease, with approximately half of these children dying in the newborn period. So what we did here was we took newborn mice, and we used a green fluorescent protein, which has nothing to do with OTC. It's just a marker protein. And basically what we were doing was we were using the AAV vector, and we were injecting it into this newborn mouse, and we wanted to see how long would it last. Because you would be worried that in the newborn period, maybe because of the proliferation of the cells in the liver, that it wouldn't last very long. And in fact, that's exactly what we found. So you could see that it got greener and greener. You know, within the first day, there was something there, and it maximized by day 7, but by day 21, it was just about gone. So, so this would not have been an effective form of gene therapy for the newborn period. We also looked, well, what happens if we re-inject the virus, but use slightly different capsids for the virus? And here, what you can see is whether we gave a single, 2, or 3 injections. When you give nothing, this is a knockout mouse that has 0 enzyme activity, just like the human hemizygous males. They all die within 24 hours. If you give a single dose, they live longer, but not that long, 2, 3 doses again. But once you start giving multiple doses of different levels, you run into immune problems. And so this was really what led us to say, at least at this point in time, the first in human trials should occur in the partially deficient patients. But we didn't want to give up on the idea of trying to develop treatment for the neonatal onset patients. And Mary can tell us how sick these children are when they come in. And this led us to the use of CRISPR-Cas9. You've probably heard about CRISPR-Cas9. This is gene editing compound. What you basically are doing, the Cas9 is an enzyme that will basically cut the DNA. And if you put in an RNA that is guided to a particular area in the DNA, you can, it's like a missile that you can direct to a very short strand of that DNA and open it up. And once it's opened up, you can then put in donor DNA in it. So you can edit the genome, you can edit out. And so we wanted to use this, and again, we went back to the sparse-for-ash mouse, the SPF-ash mouse, which has a partial deficiency. And its partial deficiency is due to a single mutation, a G to A mutation. This is similar to sickle cell disease, which has a single base pair change. So we figured, could we change this base pair back and cure the disease? So this is the OTC gene and exon 4. The exons are basically the part of the genome that comes together to express the protein, in this case the enzyme OTC. And so this was where the single problem was that we wanted to correct. So what we did was develop two vectors, because first you have to cut the gene and then you have to put in the new gene. So this first vector is the Cas9, which scissors cuts it. And then the second vector contains the OTC template with the corrected mutation. And then you just put them together and you give a single injection of these two vectors. And we did that in 2-day-old mouse pups. And then we looked at correction afterwards. And what we found was quite positive. I don't know whether, can we turn off the lights just for a moment so we can see better the, but just to give you an idea. So what we were looking at here is immunostaining again, or OTC after we had done this. Thank you very much. And so here's in the wild types, you can see a lot of staining. Here's in the carrier animal who has a partial deficiency. Here's in the untreated sparse-for-ash, you can't see hardly anything. Here we gave the vector but didn't target it to the OTC. And in these two, we did target it. And you can see at 8 weeks, we really had levels that were fairly similar to the heterozygote level. And what we had found previously was even if you just have about 20% of normal activity, you will, in quotes, cure the disease. So this was very exciting to us that, that we could now inject in the newborn period with this gene editing, and we could actually correct it, and it would be corrected for a long period of time. So we then looked further at you know, did this really have clinical significance, or was this really just staining that we were seeing? So on the left here, what we did was we gave them a protein load. You know, children who have urea cycle disorders can't metabolize protein, and so their ammonia level goes up normally. And the same is true of the sparse for mice. So this is the ammonia levels in normal wild-type animals, and this is in the ammonia levels in the untreated sparse for animals 7 days after we started them on a high-protein diet. If we gave them the untargeted gene therapy, there was no difference. But if we gave them the sgRNA, is the marker for the gene editing, the levels were significantly lower. Furthermore, when we looked at their survival rate, the wild type is up here, And so is the sgRNA. It had identical survival rates, whereas and much higher rates than the untargeted or the untreated animals. So that made us feel good. But the problem was that that OTC deficiency has 400 different mutations in families, and we were just correcting one of them. So the idea of being able to create 400 different gene therapies, didn't make any sense. So we then went back to the drawing board, and we actually said, okay, well, can we edit back in the actual complete gene itself? And the advantage of that would be that we could inject it, it would be incorporated into the cell in the liver, and as those cells divided, they would still have this new OTC gene in it. So the advantage of this would be that it would be broad, it wouldn't be mutation specific, and could be used for all OTC, and it would be long-lasting because it wouldn't be diluted out. It would just continue to proliferate. So this is what we did. So for vector 1, which was the Cas9 scissors, we used exactly the same vector. We wanted to open it up, but for the second vector, we actually put the OTC gene in it, not just try to deal with a single mutation. And so it has, again, the promoter, the TBG promoter that gets it into the liver as it should be. And so then what you've now done is incorporated into exon 4, which is where OTC ought to be, a new OTC that would be effective. And we did the study to look at what happens both in terms of immunofluorescence and enzyme activity. And we found the same thing that we did previously in a very positive way. Can we turn the lights down again if possible? Wild type had a lot of activity. The sparse spur mouse had no activity, and the targeted at 3 and 8 weeks showed really good activity compared to the untargeted or the untreated animals, both in terms of the immunofluorescence for OTC and for the actual enzyme activity, showed exactly the same thing. Pretty good enzyme activity compared to the wild type. So, so we're quite excited about, you can turn the lights back on, the idea of using gene editing in terms of gene therapy for neonatal onset OTC deficiency. So we'll see how the clinical trial goes in the partially deficient patients using the conventional gene therapy with AAV, and if that's successful, then our next step would be to work on it in neonates. This just shows you the same thing that with the gene editing of the entire enzyme, you can lower the ammonia level and improve the survival, so that it's the same as for the wild type. So what's happened in terms of the rebirth of gene therapy? We now have improved viral vectors, especially AAV, that I think will lead us forward. The improved economy will fuel investments in this area. Almost every day I get called by a venture capitalist who wants to talk to me about gene therapy and investing in that. I don't talk to them, but not unless Kurt tells me I should, because I always listen to Kurt. And the third is that there are new gene therapy platforms like the one I described, the CRISPR technology, which I'm pretty sure will win the Nobel Prize in the next couple of years. That's my prediction. There's also RNAi, RNA inhibition, where you can actually turn off a gene, and that could be very helpful for rare diseases in which there's overexpression of a particular gene. And we're also waiting the results of trials. There are a number of trials on hemophilia A and B, on spinal muscular atrophy, and on leukemia that are going right now. And if those are successful, I think there's going to be an explosion of what happens in gene therapy. And so that's kind of where I think the future is. So I'd like to leave you with some life lessons about this. So first of all, I can't overemphasize the importance of performing a clinical trial with the utmost of care and compliance. When we do less, we are not honoring our patients, and we could lead to devastating errors like what happened in my case. And I can't overemphasize the importance of our CTSA and its training for all of us who perform clinical trials. The second is to acknowledge and learn from one's mistakes. I think it's especially important for someone in my position to acknowledge mistakes and to correct them and to learn from them. But all of us are going to make mistakes sometime in our career, and the most important thing is to learn from them and to share them with those who are younger than us so that they don't become devastated and then something like this happens to them. They know that they can live through it as I did through mine. The third is that science, medicine, and humans are resilient. So even when something like this happened, and that for a decade gene therapy was dead, it was reborn like the phoenix. And I was as well. This was my first year here, and I wasn't sure there would be a second year. But there's been almost 20 now, and one can live from that. And lastly is that supportive families are essential in these times of crisis and difficulty, and I certainly am very grateful to mine. And my last slide is from my granddaughter, Rebecca, who a couple of years ago I was explaining to her my error, and she drew me this rainbow, and she wrote on it, "It's never the end of the world when you make a mistake." Thank you. I'd be glad to take questions, and I'd particularly be interested, Scott, if you might say something about what you think the future of gene therapy is going to be, given your knowledge.
Well, first of all, Mark, this was a wonderful lecture, and thank you for this, because it was not only great science and the prospects of really curing patients, but also your example of going through the trials and tribulations of a really horrible outcome that could actually affect any of us. You know, we're always on the edge. We're always looking for the changes, and even for those of us who are extremely careful, and I know you are extremely careful as a person, bad things happen, and you just showed really resiliency on how you approached this, and very thoughtful. And I think this is a good testament for the institution here. You're leading by example, so thank you for that. With regard to the area of gene therapy, you probably know that I actually am a chairman of a gene therapy company that's it's actually working on ocular diseases. So I've been doing that since 2002. This is not an area that my company is focused on, but I saw the value of gene therapy, and I knew that there was going to be a time that it was going to get reborn. And the company was formed in the wake of the incident that you discussed, but I knew the science was solid. I saw the prospects there, and obviously there's a lot of learning in terms of, as you described, new vectors to be used, obviously the whole CRISPR-Cas9, and obviously all the other new enzyme systems that are now being implemented. There will be time, it'll take time to implement these things, and obviously from a regulatory and a safety standpoint, it's going to take time to get through these, because as you described, we can imagine the prospects of success, but there are so many things that can go wrong here, and we obviously have to impart a very close focus on the safety parameters of these things. So I think you pointed out quite well, there's hope in the area of hemophilia, sickle cell disease, areas of obviously a lot of orphan diseases that people are going, affecting the liver, affecting the eye, affecting the central nervous system now. And so I think for the next 20, 30 years, this is going to be one of the hottest areas of growth in the biotechnology industry. And I'm glad you persisted in your pursuit of curing urea cycle disorders, because it obviously would be wonderful if this can help these patients. So I'm a big proponent that this is going to be successful. There is, though, let's put it in perspective. So you had, and I'll get to a question now after that long-winded statement. Is in what you learned about the outcome on Jesse, and you described it as an inflammatory disorder that was really a function of an immune response to the capsid. Are there additional lessons to be learned here about the specifics in terms of the immune response that occurred? Did he have preformed adeno antibodies that helped to promote a particularly exuberant immune response? Are there particular cytokines that were actually induced that caused the liver damage? As you are quite well aware, in our current therapies for cancer, giving CAR T-cell therapy, where adoptively transferring T-cells, you can provoke patients that really actually a number of patients now have died from, in some cases, a life-saving therapy, but we have the control of how do we do this. And we've learned that, intervening by giving certain anti-cytokines and things, but if what we know now, could you have intervened earlier if there were antibodies, anti-IL-6 and other things, that you know the mechanism a little better in terms of this disease process?
Part of the protocol was to check to make sure that there were not antibodies against the adenovirus before infusion, and he did not have that. And there was a specific pattern of IL abnormalities that we went back retrospectively to look at other patients, and in fact, all of the patients had levels. He just happened to have been the one where it tipped over, but it was not something that people had recognized or had seen in the animal models before that. And I think that also points to the importance that mice are not men, and that the things that we learn from our mice work are not necessarily transferable, because the dose also that we found was very effective in the mice had virtually no effect on the humans.
And actually, I'll add an additional comment to that. In our experiences, for instance, of giving these immune therapies, either active or adoptive immune therapies, monkeys are not men either, because it turns out that the safety profile that we've seen for many of our therapies that induce a very exuberant immune response, you don't translate from a dose effect in the monkeys to that in humans, and humans, in fact, tend to be almost more sensitive at much lower levels.
Thank you. It is 9 o'clock, so I think I will call it to a close. But any of you who have additional questions or would like to talk to me, I'll be glad to talk to you all quicker.
One comment. You said you did whatever I said, so I want to take the privilege of, first of all, to the group that put this lectureship together. Thank you for the contributions, and Mark, a stellar, in a way, an expose, but creating a sense of vulnerability and self-introspection that's rarely seen, and it, I think, is so meaningful. And thank you. I also want to link it to, there were others involved that were probably really impacted here, and I'm thinking of the nursing staff that probably cared for this young man that it probably created a lot of conflict and a lot of by the whole team. And the reason I'm saying that is that this is National Nursing Week, and so we want to be sure that we all reach out to our colleagues. There are some nurses here. I see at least 2 of our nursing leaders right there. Martha and Linda. Would you stand so everybody can give you a hand for all you do every day? But for all of us who take care of patients, you know, when you're taking care of sick patients with these types of diseases, things happen, and we have to pull together as teams to get each other through that so that we can come back and impact the lives of children and families in a positive way. And Mark, you've certainly shown the leadership for all of us to take that on in such a positive way. So thank you so much, and have a great Nurses Week. See you.